everyone. This is part 2 of the chapter 1 video. The first one cut off, so anyways, let's just start where we left off earlier. So, part 2, distinguishing between two major types of cells. So here we have a prokaryotic cell and a eukaryotic cell. So prokaryotic means before a nucleus, eukaryotic means it has a true nucleus. What does this mean? Well, a nucleus is a very large organelle that eukaryotic cells have that stores all of our genetic information, our DNA. Whereas a prokaryote does not have this nucleus, and the DNA is just kind of free floating in here in the cytoplasm. Cytoplasm is cell fluid. So eukaryotic, true nucleus, prokaryotic, no nucleus. Basically, we are eukaryotes. So prokaryotes would be bacteria, and eukaryotes would be animals, plants, fungi. You got these cells, so you got the eukaryotic cells is the easiest way to remember that. So here is an example of a eukaryotic cell right here, this big one, and then we have this little prokaryotic cell. This big eukaryotic cell has this very large nucleus that contains DNA, and the nucleus is going to be the largest organelle. And then a prokaryotic cell, as depicted, does not have this large circular feature that has a nucleus. The DNA is just kind of floating around, roaming around in here. These are other little organelles, so I just talked about the nucleus being an organelle. The cell has organelles like our body has organs, is the easiest way to remember it. Organelles are just parts of the cell that perform specific functions, which we will talk about more in a later chapter. So the eukaryotic cell is more complex, and it contains organelles that I just talked about. And then the nucleus is the largest. All cells use DNA as the chemical composer of genes. What are genes? They're units of inheritance that go from parents to offspring, to their offspring, to their offspring, and so on and so forth. And the language of DNA contains just 4 letters, A, G, C, T, adenine, guanine, cytosine, and thymine. We will talk about these a lot in our DNA chapter, so I will just touch on them. This is just one side of the double helix ladder that is DNA. So the other side of the ladder would have responding DNA. So adenine and thymine would be together, so it'd be AT, and then cytosine and guanine would fit together like puzzle pieces, CG. It won't be TG or TC. It would be like that, and then this ladder would twist and give us our double helix. But again, more on that later. And then just a vocabulary word is genome, all DNA or genes in a single organism. That's something that we did as Homo sapiens, oh my goodness. Homo sapiens is we created this human genome project, and we looked at all of the genetics of humans, and that's publicly available if you're interested in learning more about the Human Genome Project. Life in its diverse forms. So we are very diverse. As of right now, we have 1.8 million species and that we know of. Estimates range from 10 million to over 30 million species. 30 to 200 million species. There's so much of the world that we haven't discovered yet. And as scientists, we are just trying to further our knowledge in all these different areas. But here's a little chart of high diversity, medium diversity, low diversity, and here you can see the different levels we were talking about earlier, how there are a lot of primary producers, less so herbivores, and then less so carnivores. It's just that flow of energy again. This is a good little picture of how everything works. Low diversity, self-explanatory. You can see how there are only a few different species in each level, and in high diversity you have a lot of different ones. So just depending on which area you're looking at, you will see a lot of different things. So biodiversity is just so vast. This example right here is just a lot of different species of butterflies. Who knew, right? Taxonomy is the branch of biology that names and classifies these species. Binomial nomenclature, we'll talk about that later, where we talk about genus, species, is part of this. There are three domains of life. We have bacteria, archaea, and eukarya. So like we said, we are part of eukarya. We are over here, kingdom Animalia. But what are domains, bacteria, and archaea? They were actually deemed to be the same at one point, but we have recently split them up into two different domains because archaea have actually been shown to act more like eukaryotes than bacteria, so we had to split those babies up. Bacteria we're used to, we get bacterial infections all the time, but what are archaea? Different things like extremophiles are archaea that can live in hot springs, very, very extreme locations, which is why they were given the names extremophiles, like little organisms that can survive huge temperature ranges that bacteria couldn't survive in, and that we, as part of domain eukarya, couldn't survive in. They are both prokaryotes. That is something that links them together and why they were grouped together to begin with, but through research we've deemed them to be different domains. In eukarya, the domain that we're a part of, contains protists, plants, fungi, and animals. Fungi, fun fact, actually act more like animals than they do plants, which is very interesting. And then protists are our single-celled eukaryotes. Everybody's heard of malaria. Malaria is actually caused by a protist that is transmitted by a mosquito. The protist's name, I forget, but I'm sure it'll come to me whenever we talk more about protists. Underlying the diversity of life is a striking unity, especially at the lower levels of structure. So, what does that mean? That means that all living organisms use DNA. DNA, genetic information, that is what gives us life. Not necessarily gives us life, but without DNA, I wouldn't even imagine how it would work. Anyways, moving on. Evolution accounts for this combination of unity and diversity. So now we are getting into evolution. An example, the universal architecture of eukaryotic cilia. So this little paramecium has cilia, and then here's a cell from a fallopian tube that has cilia too. This is just an example of how vastly different these two organisms are. So cells from a fallopian tube to a cell from a paramecium, but how similar they are in structure and function. So now to move on to our evolution theory of evolution via natural selection step of biology. Here's a very famous quote by Dobzhansky. Nothing in biology makes sense except in the light of evolution. We'll be talking about evolution a lot in this class. Scientific evidence points to evolution, and we have not found scientific evidence that doesn't support evolution. Therefore, evolution is a theory that we learn and study a lot in biology. We won't be talking about religion, but I will say most people who are religious accept evolution. It doesn't have to be one or the other, so that's just a disclaimer. Whatever you choose to believe, it doesn't have to be mutually exclusive. So the history of life is billions of years old. Fossils document this history. I really like talking about fossils because they do provide us with a lot of history, from dinosaurs to trilobites and all kinds of other different fossils we've discovered over the years. And here's just a little phylogenetic tree that shows you what we mean when we talk about evolution. Here's a little picture. So here we have some bears. They all came from an ancestral bear 30 million years ago. 30 million years ago is hard to wrap your minds around, just like maybe $30 million, unless you have that in your bank account. If you do, good for you. But yeah, 30 million years, $30 million is just, it seems like something that's really difficult to grasp. But anyways, all of these different bears diverged from a single ancestor, so this, we'll call it this ancestral bear. And about 20 million years ago, it branched off into two different types of bears. So this is one, this is the other. This one kind of looks like it stayed the same throughout all the years and was this giant panda, whereas this guy decided, hey, let's diverge a little bit more. So here, about 10 million years ago, we branch off again. One branch goes to the spectacled bear, and the other one booms into all of these different bears, just with all these branches. One is over here and then separates, brown bear versus polar bear. But anyways, you get the idea. And this is just a little tree that shows you how we would track that. Then we have Charles Darwin in his publication called The Origin of Species. He didn't discover evolution, but he pointed out the mechanisms that happen, and the origin of species by means of natural selection is something that we look at, respect. It's just a really important piece of history and an important piece of document in the science community. Another person, Alfred Russel Wallace, actually around the same time published another work. So it's funny that they both decided to point out the mechanisms of evolution around the same time, 1859. But we're going to talk about Darwin here. So like I said, Darwin didn't invent or discover the theory of evolution. He just pointed it out. He discovered the mechanism by which it could occur. A lot of scientists had already come to the conclusion that evolution was occurring. If we look at Aristotle, you know, he was talking about differences in animals and seeing different things, etc. But what Darwin's book did is it made two big points: descent with modification, so change over generations. Change isn't going to happen immediately, and it's not going to happen like, hey, I like this characteristic, let's keep this characteristic. No, descent with modification just means that the best let's say natural selection and descent with modification go hand in hand. It just means the best characteristics suited to that environment will survive through generations, and they might modify to be a little better suited over generations, so over time, suited to the environment. Darwin went to the Galapagos Islands, and he was really struck by the diversity there. So what he saw was this geographic barrier, biogeography, which is something else that we'll talk about this semester, and how this barrier separated these species, made them develop such different characteristics. And what he specifically saw were 14 different species of finches. Let's just look at the two extremes right here. We see this large ground finch, huge beak, probably really great at cracking nuts and using that as its main food source. Here we see a gray warbler finch, the smaller pointed beak, which probably meant that he ate little insects from trees. And he saw these on all these different islands and how these beak shapes differed with the different food sources that were available. So through generations, the beaks just probably, started out somewhere here, a potential common ancestor, this size, and with these finches living in a more seed-rich environment, their beaks got bigger and developed to be better at cracking seeds open. And then the ones that were more on islands with more insect bearing, the beaks took on a different shape. But basically these beaks adapted to suit their environments over generations. Like you see here, this is a common ancestor, and then generations had to pass. Oh my goodness. Here we go. So Darwin synthesized the concept of natural selection from two observations that were neither profound nor original. Other people had pieces of this puzzle, but Darwin saw how they fit together, and he wrote his findings in The Origin of Species. He saw how they could have fit together with this common ancestor and how they diverged to create all these different species of finches and all these different islands as they got further from the mainland. All right. So some facts. More babies are born that can survive, and their survival is not random. Another fact, individual variation, individuals differ. So you're not going to see clones. You're going to see different variations among siblings, parents, others, etc. But basically the biggest inescapable conclusion here is unequal reproductive success, which makes sense when you think about humans too. Not everybody will reproduce. And that's about that. We'll talk more about this during the evolution section, so I don't want to get into too much detail. But anyways, this unequal reproductive success is what Darwin called natural selection. So the ones that are better suited to their environment will survive to reproduce, to have offspring that will be better suited to their environment, etc., etc. The product of natural selection is adaptation, so like we saw in the finch beaks. So let's get these playing. Okay, so natural selection is the mechanism of evolution. So here, oh goodness, there we go. So here we have dark beetles and lighter colored beetles. So this population obviously has varied inherited traits with these lighter ones and these darker ones. This bird is picking off the lighter ones because it can see them better. So they're getting eliminated. So these white ones are getting eliminated, they're getting eaten because they're more visible to its predator. So so with those being taken away, these survivors are reproducing, and who do the survivors happen to be? The darker ones. So the ones that the bird can't see as well. They reproduce to create all of these offspring that are also darker that the predators won't see as well. So increasing frequency of traits that enhance survival and reproductive success. So that's one. As we get into evolution though, we'll talk about the evolutionary arms race. As prey adapts, the predator is too. So in this example, the white ones are being picked off, the dark ones are reproducing to create more dark ones. Now, predation isn't such an issue. However, with evolutionary arms race, as these beetles are developing better traits for survival, so are the birds. The birds we'll probably develop eyes that can pick out the beetles more effectively, even if they're darker, no matter, that their coloration. Anyways, more on that later. So let's talk about artificial selection. So artificial selection is done by us, and it's the selective breeding of domesticated plants and animals. So here's flower example, different veggie examples, but one example that I will talk about are these dogs. So all dogs come from the same wolf ancestor about 40,000 years ago, but we came into the picture and started breeding all the friendly dogs, basically. So we removed that alpha hierarchy that wolves have, because we don't want alphas as our pets. We want the friendly, go-lucky, happy dogs that we call our best friends. So that is one area of artificial selection. Something that also happens with artificial selection that isn't so good is purebreds. So inbreeding to create purebreds, and that just opens up for a lot of genetic problems, which we will get into again some more when we talk about evolution, artificial selection, and inbreeding. So what I wanted to touch on was antibiotic resistant bacteria and natural selection. So when penicillin first came out, it wiped out bacteria like no other. Penicillin is not as effective anymore because these bacteria got resistant to that type of antibiotic, so they started getting stronger. Basically the moral of this story is take all of your antibiotics, people. If you feel better and you stop, you've only removed the bacteria that's easiest to kill because of these antibiotic resistant bacteria. If your doctor has prescribed you 10 pills to take over a course of 10 days, take those 10 pills over the course of 10 days. I've had friends with strep throat, and they stopped taking their antibiotics because they were like, well, you know what, it doesn't hurt anymore, I feel better. And then they get it again, and they're like, why did I get strep throat again? It's like, well, because you never actually got rid of it. At this point, they've infected their family, they've infected their friends, and so on, when they could have completely avoided the situation by taking all of their antibiotics. Yeah, that was just a little fun story to throw in there. So again, Darwin's publication fueled an explosion in biological research, with evolution as its unifying theme of biology, which is why we're going to talk about evolution a lot. It's saying that it's unifying, everything's related, and we can look at these relations by studying animals, which is awesome. It's not very ethical to breed humans for the purpose of science, but we can do that in mice and flies and find out a lot of genetic information, which we still are. A couple of my friends are actually doing Drosophila, which are fruit flies, genetic research at Western Kentucky University, and are still learning new things about genetics that can, benefit us in the future. All right, so this is a pretty funny story that you should probably take a second to read and pause the video. Yeah, and that's all I'll say. All right, so process of science. Science is from a Latin verb meaning to know. Knowledge is key. The science is a way of knowing. It's developed from our curiosity about ourselves and the world around us. It's been around forever, from Aristotle to Plato to all of those big names that you think of. They were all people who had a natural curiosity about the world around us and what's happening. Science can never prove a statement. It can only support or fail to support it. So I know I put disprove here, put disprove so it would mean fail to support, but don't ever use prove or disprove in science. We use support or fail to support because there's always someone that can come out of the woodwork, find something that completely goes against whatever statement you made and essentially fails to support it. If you disprove something, it just sounds so much worse. So just use support or fail to support, not prove or disprove. So all organisms are made up of cells. That is cell theory. And in the book, it says that the test either supported the hypothesis or failed to support it. Obviously, it supported it because we're still saying that all organisms are made up of cells. Do we know that? Have we tested every single organism? No, we haven't. But if an organism that is alive comes around and happens not to be made up of cells, then we'll know that that statement, you know, hey, this just failed to support that statement. All right, then we have our scientific method. So we make an observation and we state that observation, that question, the problem that we see. We gather information about that problem and then we formulate a hypothesis. So a hypothesis is an educated guess. If you hear somebody say, I have a theory, that's wrong. They actually have a hypothesis. So next time one of your friends says, hey, I have a theory, stop them right there and say, nope, you do not have a theory. You, sir, have a hypothesis. But anyways, you form a hypothesis, you test that hypothesis, look at its data, and you draw some conclusions. The conclusions that you draw will either have supported that hypothesis or not supported it. If the hypothesis was supported, then you repeat this test, this experiment, several times. If it wasn't supported, then you go back, you modify your hypothesis, you change something about it, and then you retest and reanalyze data and redraw conclusions. If your hypothesis was supported hundreds to thousands of times, then it can become a theory like cell theory and the theory of evolution by natural selection. Alright, so here we have a graph, the effective temperature and distribution of flatworms in a stream. So here I just wanted to talk a little bit about independent, dependent variables and regression lines. On the x-axis, so down here, x-axis is our horizontal axis and y-axis is up here. So the y has a little stem that goes up and down, so you remember that the y goes on the up and down axis. The x-axis is our independent variable. Our independent variable, like temperature, doesn't depend on anything. And I know using part of the word in a description isn't a very good description, but everybody basically pretty much knows what independent and dependent means, so I won't go too much into that. And our dependent variable, which is flatworm numbers here, depends on, we're saying that it depends on the temperature. So at 10 degrees Celsius, we see about, what is this, 40 flatworms. Yeah, let's say we see about 40 flatworms, 10 degrees. So all these little data points are telling us what's happening. So at 6 degrees Celsius, we see 80. At, what, 6.5, we see 70. And that's what all of these little data points are telling us. After you plot these points in a program like Excel, you create this line, this trend line. And it shows the general relationship between these points. This one shows a negative trend line. So we see as temperature increases, the number of flatworms decreases, which we would say is inversely proportional. So as one goes up, so temperature goes up, the flatworms go down. So that is a negative relationship. If flatworm numbers went up as temperature went up, we would see a positive relationship, and the line would be going this way, and the data points would be around that line. Let's move on to our squared value. So we use an R squared value again, which the program that you're using will generate for you as immediately whenever you generate this trend line, an R squared value will pop up. This tells us how close the data fits to our trend line. 1 means perfect correlation. So if this number was 1.000, it means we are perfectly correlated, and all these data points would be on this line exactly. If this R-squared value was 0, the data points would be completely random and scattered. This line would not look like this, and there would be no correlation. But because ours is very close to 1, so we have a 0.9623, we see that our data shows a negative correlation pretty strongly. So we have a very well correlated data. So as the temperature goes up, the number of flatworms goes down. Now, this controlled study, if we were to do this test again, so the flatworms and temperature test, we would want to do a controlled study of, flatworms at a normal temperature to make sure that they're not dying because they're sick. We really want to take into account every piece that could go wrong. Like Murphy's Law, anything that can go wrong will go wrong, so you want to take precautions so that nobody can come around and say, hey, they were actually sick and that's why they all died, and that just happened to look like it was correlated and without this controlled study. But with a controlled study, it's like, hey, no, we actually know that they weren't, and we used flatworms from the same population. So a lot of people find the scientific process very frustrating because, at one point we say this, now we say this. Again, that goes back to, developing hypotheses, running an experiment many, many times, and then somebody can run it one time and find something the opposite that you found, which, fails to support your hypothesis. But again, it's important for scientific discovery and everything that all of these things happen so that we can come to some kind of knowledge about what's happening around us, about what if that's a new medication. What if somebody comes around and does this trial run with a medication and finds something very horrible that could go wrong? We want to test things hundreds and hundreds and hundreds of times. And before studies are published, they have to go through peer review. So it gets randomly sent out to people with very similar credentials or higher credentials than the person that did the study, and they look through what you wrote about and edit it and say, hey, okay, everything was done well. Nobody's going to publish something willy-nilly in a respected journal. Everything goes through a peer review process and gets edited a lot of times before it gets published. So what is a scientific theory? It's an attempt to explain facts and integrate them with overarching concepts. So a good theory will be tested over and over and will never be proven, there's this word again, wrong. But a good theory will be tested 100 times over, and it will only find evidence to support it. So like cell theory, like evolution, atomic theory, heliocentric theory, or just some other ones. How is a theory different from a law? Well, a law describes patterns that are seen in nature and will have usually an equation that goes with it. So you talk about laws a lot in math and physics. So like the law of inertia, an object in motion tends to stay in motion, and an object at rest tends to stay at rest unless acted upon by an outside force. And then you have law of gravity and all other types of laws. So that is the end of our first chapter.